shall we start, madam?
So, few more minutes. Shall we wait, sir? Please.
Yeah. Just you intimate me.
Yeah, sure, sir.
Okay.
Shall we start, madam?
Yes, we can start.
Can start. Sir, Professor Sir Girish Pathade, sir. So we can start the program, sir, because 200 plus participants have joined the program.
Am I audible?
Yes, sir, you are audible, sir.
Shailaja madam, please you start the program.
Yeah, sure.
Should I share the screen or should I wait?
No, sir, I'll start the program right now. So
Right.
we'll proceed.
Okay.
A pleasant good morning to one and all present here. So myself, Dr. Shailaja, I am Assistant Professor, Department of Mathematics. I extend a warm welcome to one and all present here to this 5 days virtual faculty development program on innovative teaching and research in science, exploring the new horizons. Science is a beautiful gift to the humanity. We should not distort it. These are the sayings by our president and scientist, Dr. APJ Abdul Kalam ji. So we live in a society which is explicitly dependent on science and technology, which is hardly very less percentage of people are aware of it. That is why our former president and scientist, late Dr. Abdul Kalam ji, has said these words. So for our present scenario, UGC has recommended to do research in all for all degree college teachers in their respective fields. To meet today's requirement of college teachers, we have an eminent speaker from Pune, Maharashtra, Dr. Girish Pathade, sir. Sir, so it gives me an immense pleasure to welcome you for today's session, sir. So I extend a warm welcome to you, sir. So welcome to Dr.G.R.Pathade, sir. Namaste, sir. So who is going to enlighten us on writing on the topic, writing and publishing research papers and project proposals? On behalf of Science Forum and of Government First Aid College, Vijayanagara, so I once again extend a warm welcome to you, sir. So I would like to welcome our beloved principal, Dr. Chandrasekhara B. So who is the backbone of all these programs of our college and is very encouraging and supporting person. So welcome to this program, sir.
Thank you.
I welcome Dr. Shubha, dynamic and active IQAC coordinator of our college, and the organizing secretary, Professor Shubha from the Department of Chemistry. So I welcome you, both of you, madams. So, and I welcome all participants across the nation and also from the abroad who have joined and who have encouraged us by joining this virtual online FDP. So my heartfelt welcome to each and everyone. So who has joined this fifth day of virtual faculty development program. So I also extend my heartfelt welcome to my dear colleagues from the science stream who have put a lot of effort in conducting this program. So I once again extend my heartfelt welcome to one and all present here. So now it is over to Professor Ratnakumari B.M. to introduce today's eminent speaker, Dr. G.R.Pathade, sir. So over to Ratnakumari, madam.
Unmute. Ratna.
Yes, I'm sorry. Good morning, everyone. I feel extremely honored to introduce our today's resource person, Dr. G.R.Pathade, ex-principal, Haribhai V Desai College, Pune. He is a multi-talented, multitasking, versatile personality and also a renowned professor, researcher, and a good administrator. It's a great privilege to have him as a resource person on our fifth day of virtual faculty development program. Dear friends, I would like to give a highlights of Sir's CV. He has a number of degrees and laurels to his credit. He has obtained his MPhil and PhD in microbiology and has DMLT, CCCO, DLIT to his credit. He has 40 years of teaching experience to undergraduate and postgraduate students of microbiology, biotechnology, and environmental sciences. As a researcher, has 37 years of experience and has done extraordinary research work. To his credit, he has 150 research papers published at national and international research journals. Till date, 15 PhD students have been awarded PhD under his guidance, and 15 students have already completed MPhil degree under his guidance. He has successfully guided 200 UG and 70 PG student projects. Sir is ex-chairman, BOS Microbiology, SPPU Pune. He is ex-members of Academic Council, SPPU Pune. He is also a member of BOS of Shivaji, Solapur Krishna University, and he has written 55 books in microbiology, biotechnology, and environmental sciences. He is awarded with 5 Indian patents for his excellence in research work. He is ex-senator of Shivaji University between 2000 to 2005. He has won 3 awards at national level and also 3 awards at state level. The first national award as the best teacher in 2011, Delhi, and second award as an environmentalist of the year 2017, Ranchi, Ecological and Environmental Society, Kolkata, and third award as an environmental microbiologist of the year 2017, Confederation of Indian Universities in New Delhi. He has also worked as chairperson in an international conference at USA, worked as a PhD referee for Mumbai, SPPU, Shivaji, BAMU, YCMOU, CSU, Kanpur, and Rajasthan universities. With this brief introduction, I request Dr. Pathade sir to take over the session. Over to you, sir. Thank you.
Thank you. Thank you. Thank you. Screen is visible?
Yes, sir. It is visible.
Okay. I'll start my session. In between, if any problem comes, please you contact me on phone.
Yes, yes, sure.
Good morning, one and all. First of all, I express my sincere thanks to the organizers, the principal of this college, my learned colleagues, Shubha Madam, Shailaja Madam, Ratna Madam, and other learned colleagues, all the participants, delegates who have participated in this particular session. I congratulate the organizers for organizing this 5 days virtual faculty development program and introduce various eminent personalities and their talks as a exposure to the faculty members. I welcome here all the faculty members who have participated in this particular session. I'll talk on the two aspects. When I was talking with the Shailaja Madam, Shubha Madam, she told that I should talk on the research paper publication process as well as how to write project proposals. So two aspects are there. I'll first talk on the how to go for the research papers, how to publish research papers, and then after that I'll go for the project proposals. Now everybody from the scientist wants to publish his work in the form of research articles, and he always wants to publish in the good journals. We say that the journals with the good impact factors, and for that purpose, many times we send the research papers, research articles to those journals, and sometimes we find that a paper is rejected. Now, why this thing happens? For that purpose, it is very, very important to know that what are the exact components of the research paper, how it is an art, how to write. It is a skill, how to write the research paper. And in that, the scientific writing is very, very important, especially the scientific English, that is very important. Scientific language is very important. So I'll discuss all these aspects, how to go for the research paper. Now, when we talk about the research, it is an art of scientific and systematic search of for the knowledge. There is a path from known to unknown. What is the research paper or research article? It is a written and published report describing original scientific research results. Now, about the research paper or research article, how to write it. Now we know that many research workers they like to work in the laboratory, but they don't like to write. Scientists must not only do the science; he must write the science. Again, very important. Just I told that paper can be rejected. Your good paper, good work is rejected. Bad, that is because of the bad writing. Bad writing can and often does prevent or delay the publication of good science. Generally, more emphasis is given on the technical aspect. When we write the research article, we give emphasis on the technical aspects, and the communication arts they are neglected. So care must be taken with respect to that. Many good scientists are poor writers. This thing happens. Many good scientists are poor writers. Perhaps many scientists don't like to write. Scientists must write. Hence, it is need of scientists to undertake a formal course in scientific writing. You should have the training of the scientific writing for developing high probability of acceptance of manuscript for publication. This for your acceptance of the paper, your scientific writing skill is very, very important. Now the publication, this is publication is a goal of the research, and this is a process. Now in this process, what is needed? Why we should publish our research work? It is very, very important. Many times people they don't publish. This excellent work is there, good results are there, our aspiring results are there, but they don't publish. But that is very wrong. You should publish. Why? What is the significance of publication? Publication is a goal of scientific research. The scientists are not measured. That is very important. Scientists are not measured by their skill in the laboratory manipulation, not by knowledge of scientific subjects, not by their charm, not by intellect, but are measured and become known by publication, or remain unknown if not published, become known by their publication. A scientific experiment, no matter how spectacular results, is not completed until the results are published, and is the cornerstone philosophy of science, as it automatically adds to the existing scientific knowledge. It is very important. The scientist must publish. Otherwise, we know that if it is not published, if your work goes in the hands of some other people, he may get credit. This thing has happened in the science. We know so many examples are there, and for that purpose, scientists must publish his research results. Now, so what is scientific writing? The purpose of scientific writing is to communicate scientific findings. The scientists must learn to use the English language, because we use English language because it is a language of communication all over the world. Most of the countries, they or the scientific world uses this, and for that purpose, this English should be used with a precision. The best English is that which gives the sense in the fewest short words, and which conveys message in minimum short words. So in scientific writing, research paper writing, that is very important. You should precisely present the language, and in the minimum possible words, few simple words, you should present it. The scientific writing involves clear, simple, and well ordered words without much ornamentation. People, they have a tendency to go for ornamentation, but don't you use ornamentation. Simple possible words should be used while presenting scientific problems, experiments, findings, and conclusions. In other words, scientific writing demands absolute clarity. There should not be any confusion in the language which you use. Now, publication of research paper or article in good journal. Again, people, they, because it is a demand of the, now we know that it is our University Grant Commission, and the CARE list is there. And in that list, the journals are enlisted, where impact factor is mentioned. High impact factor indicates that the good quality of the paper and journal is there. So the researcher always wants to publish his paper or article in the good journal with the high impact factor. The scientific journals are rated on the basis of the impact factor, and this high impact factor, high rating, good quality of journal, and vice versa. Publication means disclosure. When we publish it, of the article, maybe offline. Nowadays, offline the journals are also there. Then, sorry, offline, that is a written, and online, orally in the conferences, seminars, or via ICT or recorders. In this way, you can go for the publication. Paper or article may be in the form of journal, review, conference report, posters, meetings, extended abstracts, magazines, book, periodical, new paper article, etc. Paper or article publication may be primary in the journal, or secondary means in abstract, that is it is indexed in the abstracting. It is called indexing. For example, chemical abstracts, biological abstracts. When your paper is published in the journal, that is a primary publication. Then your paper may be indexed in the chemical abstracts or in the biological abstracts, like that, and only abstracts are presented there. So one can refer those abstracts, and on the basis of that, they can get the paper. Now, this is the research process. In this research process, how to start for the research and how to go for publication. In the research process, it is very important defining the research problem, developing a research idea. First, you have to select the research topic. Now, research topic can nowadays we are very lucky that so many research search engines are there, and through search engines we can search for different topics, and you can get the idea, or you can discuss with your teachers, your guides, your supervisors, or you can discuss with your friends and experts in the field, or even you yourself keep your eyes open and look at the nature or surrounding. You can get the newer and newer ideas. Say, for example, nowadays we are facing corona problem, COVID problems, so many problems are there, and so many unanswered questions are also there. There you can find some problems, and you can select any one problem or aspect, and then one should go for the what we say the review of literature on that aspect. When you select the problem, then you have to go for the search of the literature. You will get a number of papers and articles, and then you can read those articles, and then you can formulate the hypothesis, assumptions, aims, and objectives. What to do? That you have to formulate, and after that, then you have to design the research material and methods. Which material, which methods you are going, what kind of experiments you will perform, like that. And then you will, out of experimentation, then you will collect the data. You will record the data properly. That is very, very important. Then you will analyze the data. That data analysis you will do. You will use the statistical approach also, and so you will validate the hypotheses and aims and objectives. So hypothesis testing, discussion can be, that is hypothesis testing and discussion. Summary and conclusion, you will summarize, and then conclusions you will find out of your work. Conclusions will be written, and that is in the form of interpretations or conclusions. And then you will prepare the report, that is maybe research article, research paper, or even if you are doing the PhD, you can write the thesis and like that. And then you can publish that. If it is a research paper, you can publish in the suitable publishing agency like journals. You can select the journals. Care list in our country, care list is there, or you can select any international journal also. So, and in that journal, instructions to authors, this page is always there, that you should read, where instructions are given how to, what is expected for that journal. This is the overall the research and publishing process. Then research article format. In how to prepare the research article, there is format. The logic behind the development of format for research article or paper for publication can be defined in the question of, look at what question or problem was studied. Answer is introduction. How was the problem studied? The answer is material and methods. What were the findings? The answer is results. What do these findings mean? The answer is discussion. Means your paper format, article format is organized in the form of introduction, material, methods, results, and discussion, which is called IMRAD. IMRAD acronym is a group of words. IMRAD format. So in the scientific writing, in the publishing articles, IMRAD format is the most common one. No doubt people, they have modified it slightly nowadays, but the best method for the publication is this, the IMRAD acronym or IMRAD format. IMRAD format developed in late 1900, especially after work of the Louis Pasteur, because Louis Pasteur, the great man who had made 14 outstanding contributions in biology, although he was not biologist. In the biology, biotechnology, microbiology, he is known as honorary father of biotechnology, honorary father of microbiology as well. And his all the 14 contributions were, each contribution was heavier than a Nobel Prize. He should have obtained 14 Nobel Prizes. Unfortunately, he did not get, because at that time Nobel Prizes were not there. But his work insisted for IMRAD format. So in the late 1900s, to avoid following descriptive, non-structured or chronological orders, old style of the present research paper. Old style was very simple. First I saw this, then I saw that. First I did this, then I did that. Scientific aspect was not there. Scientific language was not there. And for that purpose, IMRAD is very important. IMRAD format publication helps to potential readers to use data, and they can assess the observations, repeat the experiments, evaluate the intellectual process like conclusions and interpretation. So absolute clarity is obtained in the research article if you follow the IMRAD format. Now, how to prepare a research paper or a research article? These are the components of research article. When you have to prepare research article, keep in mind these points. Title of research paper or research article. The first, authors and their addresses. The authors of the research paper, their addresses, Then you have to write the abstract summary of your research paper. Then keywords for the objective, that is, they are very important for the indexing purpose. Then introduction section. Just a minute. Introduction section, then material method section, resource section, discussion, summary, conclusion section, stating acknowledgements, citing reference, and writing bibliography. These are the major components of any research article, research paper. Now we'll go one by one. Then how to prepare title of the research paper. It's right from title you should be very careful. When you perform the work, your results are ready. Then title. Title, remember about the title. Remember about the title. Title is the mirror or label of your paper. First impressions are strong impressions. Now, there should not be syntax error, no syntax, order of words, because this, if the order of words is changed, it can change the meaning. For example, bacteria causing mastitis are characterized by GLC. It is a wrong syntax. Syntax error is there. It should be use of GLC method for characterization of bacteria causing mastitis. Yes, while writing in English language, contents of paper, contents of paper in the fewest possible words. For example, action of streptomycin on Mycobacterium tuberculosis. Very simple, adequate in the fewest possible word. It gives the meaning. Action of streptomycin on Mycobacterium tuberculosis. Indexing and abstracting services require accurate titles. The title should not be too short, like this: studies on brucella. What kind of study you have done on the brucella? Which species? It is incomplete, or it should not be too long. They are called poor titles. So for example, isolation and identification and characterization of bacteria, yeast, mold from so and so. Such kind of long titles should not be there. They are poor titles. Do not use waste words in the titles like studies on. Commonly, it is common that people will use studies on so and so, investigations on so and so, and opening like that. Don't use such waste words, because in the journal, length of the sentence and total words is very important. Total pages counts. So it is very important. Don't use waste words. Use detailed names of microorganisms. So you have to use detailed names of microorganisms. Don't use abbreviations. E. coli, write Escherichia coli. If you want to use any enzyme, write full name of enzyme, say streptokinase or amylase, like that. Complete. Don't use any short terms. Title can be used as assertive sentence. Sometimes titles can be used as assertive sentence. For example, interleukin is required for immune function of lymphoid cells. This can be also such a title is called the assertive type of sentence or title. Running titles or heads are there. What is the running titles or heads? They are printed at the top of each page. When in the journal your paper is published, if the first the page is there, the journal title is at the left top. Title of journal, a scientific word, or a journal of microbiology at the left corner top, and at the right corner top is the title of your paper. So in this way, such kinds of the methods are used. Running title heads are there. No abbreviation, chemical formulas, proprietary names, jargons. That is the common terminologies, the common words which we use in the what we the Bolibhashi language should not be used. And alike, you should not be using the titles. Hanging titles are also there. What are hanging titles like this? Volcanoes collapses. Volcanic collapse. Relation to hydrothermal process. Then for hanging titles. Straightforward titles like new color standard for biologist. Straightforward title. This titles can be written by different ways, but remember, title is a mirror or a label of your paper. It should be precise and very accurate. Then how to list the authors? Because below whose paper this paper is, how to list the authors and their addresses. So authorship is the one who has made significant contribution to experiments and takes intellectual responsibility will be the main author. That is we always call corresponding author. Main author will be there, and his name is the first, and then names of other authors you should write. Okay. Then at the end will be the name of your guide or your laboratory supervisor. While other helping hands, the technicians are, then other helping hands are there. You can incorporate their names in the acknowledgement, but the actual research workers' name should be there in these authors. Then while writing the name of author, preferred designation is like this: first name, then middle initial, and then last name. Say Arun G. Patil, like that. This is a common. But people that they use say A.G. Patil or Patil A.G. like that. But the desired one is Arun G. Patil. Sometimes after names, degree may be written. It is uncommon, but many in some papers it is written. Say Arun G. Patil PhD, Arun G. Patil MD. But it is not common. The addresses of author are mentioned to identify the author for getting the reprints to readers from respective main author. Means if the reader wants to contact the author, then addresses are they are must. Name and address for laboratory in which work is done is given, while change address also can be given, like as a present address. If it is changed in the footnote, you can give the change address. So address of author, list of author, address of author, laboratory where they have done the work, the institution where they have done the work. When many authors are from different institutes, this thing happens. One author is from one institute, another author is from another institute, third author is from third institute. Then you have to mention that by giving some notation. We use stars, 1 star, 2 star, 3 star, like this. Unless scientists wishes to publish it, sometimes it is we read in the place of author they write anonymous. This is if the unless scientists wishes to publish anonymously, you have to, otherwise you have to give the full name and full address of the authors. Say for example, the title I have given here. First example, the antibiotic streptomycin acts on the microbial tuberculosis at high dose. If this is a title, then authors say for example Arun G. Patil is there, Jain Joshi is there, Niranjan Jadhav is there. 1, 2, 3. See, 1 star, 2 star means they are from same institution, and 2 stars means from other institutions. So first 2 of people, they are from the Department of Microbiology, say HVD College, Pune. 2 stars indicate that the Department of Microbiology is SPP Savitribai Phule University, Pune. Like that. And you should write in this way. This is the format. The antibiotic streptomycin acts on the microbial tuberculosis at higher doses. So these 3 authors' names are there. Stars are, notations are there, and then below that you are given the departments and the institutions where they have done the work. Well, second example is also there. Optimization of medium composition for aspergillus niger for its decolorizing activity of the distillery spent wash. Authors, they are there. Then you should write. So for AC College is there. 1 star is there. KK Shinde is there. 2 stars are there. They are from 2 different departments. This is the method how to write the authors and their addresses. So title, then authors, their addresses. After that, it comes the abstract. You have to write the abstract of the paper. Abstract is viewed as a mini version, summary of your paper in brief. Suggested rules are there for abstract. It should not exceed 250 words. It should be typed in single paragraph. Okay, 1 paragraph. You should not write 1, 2, 3 paragraph. Single paragraph. It should be there 250 word maximum. Many times people they write too big abstract. No, it is not necessary. 250 abstract. It is desired 250 words abstract. It should provide a brief summary of each section, as I mentioned in IMRAD. Introduction, material method, results, and discussion. So this abstract should touch to all the aspects, sections of the paper, so that reader should identify the basic contents of paper quickly and accurately. It should state principal objectives and scope of investigation, describe methods employed, and summarize the results with principal conclusions. Written in past tense, abstract should be written in past tense because it refers to work done. Never give any information not stated in the paper. The things which are not given in the paper, don't put in the abstract. And generally, references are not cited unless any modification in previous published method is done. References are generally not given. If any modification you have done in the some procedures, then you should give the reference, otherwise not. Types of abstracts. The above rules, these above rules are used for generally for the primary journals, good journals, which are the good read journals, peerly reviewed journals we call. Secondary services, chemical and biological abstracts, and are called while they are called the informative abstracts. While the abstracts, which give idea of research subject of paper and does not represent full paper, they are called descriptive or indicative abstracts. And such abstracts are used for review papers, conference reports, or government reports like that. So remember, the indexing and abstracting systems are geared. Yeah. Then keywords you have to write. After abstract, you have to write keywords. In the next slide, I will show the abstract. So indexing and abstracting systems are geared to keywords. Keywords we write after abstract. Title, author, addresses, abstract. Below that we write keywords. And these keywords should highlight the significant content of paper. The keyword should be there in the title. Preferably they should be there in the title. Hence, the right keywords must be provided in the paper, preferably below abstract and in the bracket. For example, if your paper is the antibiotic streptomycin acts on the mycobacterium tuberculosis at higher doses, then so keywords will be like this: antibiotic, streptomycin, mycobacterium tuberculosis, dose. So all these keywords almost they are there in the title, and they should be like this. So this is the abstract. Abstract written. See, one paragraph abstract is there. So that abstract is for this that is on aspergillus niger, how it is used for decoloration of spent wash and optimization of medium for that, medium composition for aspergillus niger to decolorize the spent wash. So this is the abstract. I'll not read this abstract totally, it will take time. And these keywords are the decolorization, optimization of the medium, distilled spent wash, aspergillus niger. In this way, one paragraph, hardly 250 words are there, you should write. Now, how to write the introduction section? It's after abstract, we write introduction section. So suggested rules are there. It should be written in present tense. Introduction is written in present tense. It should clearly present the nature and scope of problem investigated. To orient the readers, it should review the pertinent literature. It should state the method of investigation with a reason for choice of particular method. Why you are using particular method? Justification in brief. It should, in one sentence, it should state principal results and conclusions of investigations. A bit of repetition with abstract is often desirable. Sometimes some part from abstract may be repeated in introduction. It is desirable. The closely related references may be cited customarily at the end. Closely related reference in bracket you can put in at the end. If specialized terms and abbreviations are used in the paper, they must be defined in the introduction. So specialized abbreviation, if they are there, then these may be involved in the full form in the introduction section. So in that way, introduction section should be written. It can be written in the 2 or 3 paras, small paras. So this is the introduction section. So 2 paragraphs here they are mentioned. About the distillery, it is mentioned. About distillery waste, it is mentioned. Then what are the problems with distillery waste? How it is a strong waste, colors, strong color is there for that treatment. Which methods are there? Here references are given, and the aspergillus niger, which is used, similar organisms are used by people, and in that way, introduction, and it has covered the aspects of this particular paper. Now, how to write material and method section? purpose to describe experimental design. What is the purpose of material and methods? Is a very, very important section. Material and method section is very, very important section. To describe experimental design and provide necessary details to reproduce similar results. Material method section should be written in such a way that your results, your experiment should be reproduced by anybody. If anybody wants to perform experiments, reproducibility should be there. Then and then it indicates that your material method section is correctly written. And it is a cornerstone of the scientific method that your results with scientific merit must be reproducible. And that is very important. That much details you should write in the material methods. This section is read mainly by the peer reviewers, and on the basis of particular this section, the reviewer may recommend about acceptance or rejection of paper in spite of your hours for results. Your results are excellent, but you are written very poorly with particular section material methods. So it is very, very important. This section should be correctly written. Material. Now remember, technical specification, quantities, source, physical and chemical properties of material used must be given. If we use some chemicals and material, then you have to mention the details of that, because if your experiment is to be repeated, then it will be reproduced only when the material which you have used with specific, that should be used by the person who is going to repeat that. Use of generic names and not the trade names. So for example, lysozyme is the generic name. Don't use some names like the aristozyme, because that would be sort of advertising. So you should not use trade names. Use names which are generic names. Genus, species, strain designations of experimental animals. If you use animals, plant, microorganisms, then you should write this genus name, species name, some strains or subspecies. That should be written in detail and accurately. It should be given. In case of animals, you should give age, sex, genetic and physiological status also, because these are very important. As we know that experiments with animals, they're easy, but interpretation of results is very important, very, very difficult, because if you have not followed these aspects, age, sex, genetic and physiological properties, you may get variable results. So that is very important. When you perform experiments in material methods, when you use such kind of material, you have to give the details. If you use human subjects, if you use human beings as experimental animals, if you submit human beings for experimentation, then informed consent is very important. Written informed consent, that is the consent that yes, you can use my body for the experimental purpose. Written informed consent is very important. That should be added. That should be stated in the manuscript. Instructions to authors of the selected journal be referred for specification. So I told that when you select a particular journal, then in that instruction authors are there. They have given clear-cut instruction that how you should write the paper, what should be specification for this particular paper. That is very important. Then the methods. Remember, in the methods, methods are written in chronological order, but sometimes the methods which are related, they can be put together. The detailed methods and procedures are given unless widely known methods are there. In that case, you can write the reference. Means common methods are there, widely known method. Then in that case, no details are required. Just you can give the reference. Otherwise, if you feel that your method may not be available for the reader, you should give the details. Material methods may be presented in tabular form. You can present tabular form also. That the method used, the reference, and that is the you can first of all we can say the aspect that is which experiment you are doing. Say chemical oxygen demand. Then what is the method used? It's the title, the dichromatic effect method, and the reference like that. So you can prepare the tabular form also. And in this section, headings and subheadings with proper numbering are given, which helps further to match heading and subheading in the research section. Say for example, in the materials, if you have given the section like this, the numbering like this, say A, sample for experiment, sample collection, B, glassware, C, chemicals. In the glassware, again you will write B1, B2, B3, B4. In the chemicals, you will write the say solids, liquids, say C1, C2, C3, like that. Numbering, which proper numbering should be given. Equipment which you are using, say D1, say spectrophotometer. Then you have to mention the make, the Shimadzu, and you have to give the model, because that also matters. The D2 GLC is their Merck model. It's like that. when you use you have to give the detailed specifications. In the methods also, sample collection, B is experimental design, C analytical methods. In the analytical methods, say for example, C1 is protein estimation, C2 is carbohydrates, C3 is lipid. Like that, proper sequencing, proper numbering and sub captions you should give, and this that will help you in the results also when you write a results section. Important tips are there for this section. References must be cited for material methods. When you write material methods, you cite the references. Don't mix results in this section. Don't mix results in material methods. The only rule followed for material methods method is enough information must be given so that the experiments could be reproduced by the competent colleague. If anybody wants to perform experiment by using your data, your methodology, you should be able to reproduce your results. Best way to give the finished manuscript to your colleague when you prepare the research paper or article, give to your colleague and ask him to read and see whether he can understand the methodology easily and find out some errors or mistakes are there. This you should give to third party for reading, and then accordingly you can make the changes, or even you can give to your say senior teachers or even your supervisors also get the idea. Use precise, grammatically correct scientific English. That is again very important. No syntax errors. Grammatically correct. Even errors of comma also matters. Say for example, employing a straight platinum wire rabbit, sheep, and human blood agar plates were inoculated. Simple sentence is there. Here, comma error is there. Employing a straight platinum wire rabbit. Platinum wire rabbit. Incorrect. Here, after wire, platinum wire, comma is required. So, a simple comma also can change the meaning. So, you have to take care when you are writing. There should not be grammatical error, syntax error should not be there. Telescopic writing should be avoided. What is the telescopic writing? Look at. Blood samples were taken from 48 informed and consenting patients where subjects ranged in age from 6 months to 22 years. Here there is no grammatical mistake at all, no grammatical mistake. But this is a telescopic writing. Question is there: how 6 months old infants gave informed consent? That is wrong. This is telescopic writing, and that should be avoided. No spelling error should be there. Commonly, people they make spelling errors, especially writing the names of the organisms or the plants, animals, microorganisms like that. So there should not be spelling errors. Then material method section. This is the example. How to write material. Say organism full name is given as per as Niger. What is the so that full name is given. Incubator shaker is there. Toshniwal. The model number is given. Chemical, say analytical grade, Merck. Like that, you have to give the details. Methodology, optimization of carbon source, optimization of nitrogen source, optimization of pH of the medium, optimization of inoculum size, how much amount of aspergillus niger you are going, biomass you are going to add, or optimization of temperature for incubation, or incubation period. Is when you want to perform the experiments, then you have to write it is in detail, and you have to write the reference also for that particular method. Now the how to write the research section. It is the core of your research paper. That is the data. The results should be presented in the past tense, precise and clearly. Ingredients of research section. Number one should give overall description of the experiments. Which experiment you have done. Providing big picture without repeating the experimental details. Don't write methodology. Just title that we have done this and this kind of experiment. In brief, big picture you should give, and previously provided material method that should not be given. Second is the presentation of data. Only repetitive data should be given. Again, that is very important. If repetitive data is not there, then you should write in the descriptive form. If few determinations are there, few numbers are there, then you write in the descriptive form, in the sentence form. But if repetitive determinations are there with many variables in your results, then effective tables or graph, they are used with clear headings. Proper headings are to be given. The results may be present in the form of figures, photographs, or micrographs. You can go for cropping and framing like that. Properly you can present. Good presentation should be there. Don't repeat the results. Say for example, how people they repeat the result. If the data is present in the table form, if you have data in table form, the same data should not be put in the graphical. People, this is common tendency of people. Table is given and same table is present in the graphical form. Generally, it should not be repeated. The table gives idea about the numerical data, while the graph gives idea about picture of trend or shape of the data. So graph is preferred. Graph is preferred. Statistical tools are to be used because to validate your results, to validate your hypothesis, to properly present your results, statistical tools are to be. N number of tools are available, and these tools should be used. Statistical tools should be used. You can use various statistical methods also, approaches also. ANOVA is there, test of significance, graphs are there, t-test, chi-square test, so f-test, so many tests which are there, statistical tests are there, or statistical approaches are there. You should include so that it will consolidate your results and your data. How to write the discussion, summary, and conclusion section in the paper. Remember, it is the hardest section to write. Discussion section is hardest section. It requires clarity in your data. It requires clarity in your mind about your results. Although valid and interesting data is there, because of faulty discussion and wrong interpretations, the paper can be rejected. Although your data is good, results are good, but your discussion, if it is wrong, not to the point, and if wrong interpretations are there, paper is rejected. Essential features of good discussion. Discuss the results in detail with principles, relationships, and generalizations. Point out any lack of correlations and define unsettled points. Negative results also you should discuss. If unsettled points are there, if there are some lack of correlations, where you expect some correlation but you did not get, then you should mention that in your discussion. That matters. Sometimes people they omit the negative results which you get, but you should write unsettled points also. First discuss your results. Your results first you discuss and compare your results or correlate with previously work done by the people. Compare with there, and then by comparison whether your results match with that. If your results don't match, why? Any possible reasons like that you should discuss. Discuss practical and theoretical implications of your work. State your conclusions clearly and summarize your evidence for each conclusion. For every conclusion, you summarize give the evidence from your data. Remember, the primary purpose of discussion is to show the relationships among observed data, observed facts. Discuss significance of your work. So in the results you have discussed, and in the summary conclusions, remember the discussion should end with short summary. You should summarize your whole work in say 1 page or 1.5 page, and then you write conclusion regarding to significance of your work. Need not to draw too many conclusions and don't repeat the result part. Many times people in the conclusion they write result part. The optimum pH obtained for this and this experiment was 7. The optimum temperature obtained for this and this experiment was say 37. These are the results. These are the results. Don't repeat in the result this conclusion. Only conclusions you should write. Don't draw cosmic conclusions also. Don't extrapolate the data. Your data is too short and don't extrapolate. Just you have to shine a spotlight on one of the truths, and in the simple statements you should draw the conclusions. Don't go for the cosmic conclusions. Say here the results they are mentioned. Say the medium composition for the decolonization activity of distilled spent wash by Aspergillus niger. This is the paper, and here for that purpose, for that Aspergillus niger to grow and decolorize the distilled waste. Distilled waste is a dark colored waste. To decolorize that waste, medium composition was designed here, and carbon source, different kinds of carbon sources are used, different concentrations are used, and then what kind of results they are obtained. These are the results mentioned. The glucose for glucose, fructose, glucose, lactose, molasses, sucrose. These different sugars are used with 3 concentrations, 1, 2, 3. Which is the best here? Glucose is the best. Is that the bar, the top bar indicates that the glucose at that concentration has given the good results. Like that, you should mention the results, and you should discuss, say, nitrogen source, different nitrogen sources used. For example, peptone is there, urea is there, ammonium sulfate is there, sodium nitrate is there, yeast extract, which are the which has given best results. The peptone has given first concentration, which is used. It has given, say, 0.1 concentration. It has given the best results. Then here is the pH. Say, look at the pH. The plateau is at pH 6. So that is the optimum temperature. See, 35 degrees centigrade. That is optimum. Like that, and various temperatures are used, various pH ranges are used. On that basis, the best pHs and best temperatures are selected. Say, inoculum level, how much amount of the inoculum to be added, and here it is mentioned. Say here 50%, like that. It is 5% like that. It is mentioned, and on the basis of that, variety of inoculum levels are used, and which the best results of decolorization obtained therefore. And once you optimize all these parameters, then using all the optimized parameters, then you perform the experiment and see what kinds of results you get. Say, this is the example. Photographically, it is mentioned. This is the original vest with the very dark color, and this is the after decolorization by using optimized conditions. So when you take this in the test tube, you will see the comparison. In the left test tube, you see dark color vest is there. In the right test tube, the color is almost more than 80% color is vanished. So in this way, and then you can write conclusion. In brief conclusion, I told you should not write the results. So whether your performance from carbon source, nitrogen source, or the pH or the temperature, you should not mention the glucose is the best or the like that at this concentration, or the nitrogen source at this concentration, or at this pH. No. Decolorization of spent wash by Aspergillus niger to the extent of almost 79% was there. Overall, our results indicate that Aspergillus niger could be an effective bioremedial agent that can be exploited to reduce the color of molasses best distillery effluent, particularly at places where ample amount of water is available for further dilution. But that is the conclusion. Don't repeat the result part. That is very, very important. How to state the acknowledgements. Remember, life is not so short, but that there is always time enough for the courtesy. So you should be courteous enough. So you should have courtesy. You should acknowledge to all who have helped, knowingly or unknowingly. So, for example, acknowledge technical help from your or outside lab, help in valuable discussions. Somebody has helped you. Provision of special equipments from your place or from some other institutions, or material which is provided by your laboratory or somebody else. You should put in the impressive words. Acknowledge financial support. For example, grants. You often grants, UGC, DST, DBT. From where you have got grant, from any source, sponsorship is there. You should express your sincere thanks to them. Fellowships are there, contracts are there. You should acknowledge. Express sincere thanks to friends and colleagues for help in, again, pleasant words. Express your sincere thanks to your family members as well. So that is very important. This section, it is a total right of the author to write this particular section, and he can put the proper acknowledgements in very good, impressive, and pleasant words. Then how to write the references. So this references, when you write, by two ways you can do. In the data, when we cite the references, how to cite the references, and in the reference section, the bibliography, how we enlist the references. The first here in the text, when we cite the reference, here either name, the way is the scientist name and followed by year. Say, for example, you have written one paragraph. For that paragraph, you want to write a reference, Patil and Mishra. Then you write the year, Patil and Mishra, 2008. In this way, this is called the scientist, name of the scientist and year of publication method, or a number of reference from the bibliography. Say, for example, this Patil and Mishra, 2010, this reference in the bibliography list, you say number 8. Then just you write 8 in bracket. Some other reference is there whose number is 7. Some other reference is there whose number is 18. Just you write 8, 7, 18 in bracket. Don't write all Patil, Mishra 2010, comma, then other, 10, comma, then other. That will be long. So either this first method, name of the scientist, year method, or you can write the number from the bibliography. And then when you want to prepare the list of the references in the bibliography section, different approaches are there. Many varieties of reference styles are there, but the most of the journal site references in the one of the following ways. 3 ways are there. Name and year system. Just we have mentioned Patil and Mishra, that system, where it is very popular method and convenient to author because the numbers are not given to the reference. Just name and author, that year is given. Hence, new references you can add or remove at any time. Means reference, you can modify the reference list. For example, Patil essay 1998, generation of biogas from Temple West, comma, Journal of Pollution Research. Say this is a volume number, and then ppm is page number 1051. This is the scientific method of writing the reference in the reference list. Second method is the alphabet number system. Means it is a modification of name and year. Only thing is that you have to give the number and alphabetically you have to arrange. So it is a method like this. Patil, it is a number 16. Then the 15 is the Jadhav. You have to give the number and you have to organize alphabetically. Then citation order system. First 2 methods which are there, any 2 methods can be used. But here it is written as per the order of that reference in the text. The first reference which is coming in the text, you can write first. After on the second reference is coming on the next page, you write that. As per the order, how it appears in the text, accordingly it is written. For books and pamphlets, how to write? If the book is there like this, then author is say Kothari JR Quantitative Techniques, TMH Publication Private Limited, then year, volume 1 and 2. For magazine, periodicals and newspapers, say Gupta RD Safety Measures in COVID-19. Sakal Daily Newspaper, if the newspaper is 20th April 2020, page number 6. Or for a website, you mention the websites. So use widely accepted abbreviations. Again, when you are writing the reference in the list, journal abbreviation is J, abstract is the ABSTR, bacteriology, bacterial, then advances ADV, analytical ANA, and research like RES. This accepted, scientifically accepted, worldwide accepted abbreviations you can use. This is the reference system. This is given. And that is a name and year as well as it is. This is second alphabetical, or the numbers are there. In this way, you can write the references in the list. And this is the publication process. Once you prepare the paper, preparation of paper as per the instructions to author of selected journal with good impact factor. Then you have prepared paper with title, authors, addresses, abstract, keywords, introduction, material methods, result, discussion, summary, conclusions. You have stated the acknowledgements, cited the references in paper, bibliography, everything you are written correctly. You are satisfied with the paper. Then you mail that soft copy of that manuscript to the chief editor of the selected journal. With publication fee, you have to be very careful. As per instructions of the authors, to the authors, the publication fee, what is there, you have to send, and along with your paper, you have to send the publication fees. Then evaluation of the manuscript will be done by the members of editorial board. When you submit it, then it will be circulated by your chief editor to the members, depending upon their expertise, and then they will give the PR number. That is a publication registration number will be allotted to your paper, and they will send you that PR number, so that when you want to contact, just you have to write that PR number, and accordingly you can contact them. So what happens that when it goes to the members of editorial board, then if the majority they don't satisfy with the paper, that is, the paper is not suitable for the publication, then it will be rejected, and rejected copy will be returned to author. And if your paper is to be accepted, if your paper is a very good paper, properly written paper, then precisely written paper is there, directly it can be accepted. You mention that your paper is accepted. Or if some modifications are expected, some corrections are expected, then they will add the acceptance with the corrections. Then the paper is returned to the author for corrections. And corrected paper copy, again that author will return to the chief editor. So chief editor will go through that corrected copy. He will see that proper corrections are made, and then he will get satisfied. That team will get satisfied, and that proof, that is called galley proof, will be sent to the author again. That this is the final proof. So approved, this galley proof will be approved by the author, and then it will be sent back to the chief editor, and then that chief editor will go for printing of the issue and journal in the journal, and if required, reprints will be also copies of the paper in form reprints will be there, and then this copy of journal will be sent, and reprints will be sent to your to the author, and in this way, this whole publication process is there, and this process may take 3 months to 6 months, depending upon the impact factor and the type of journal. So this is overall the how to write the research paper, research article, and how to publish research article in the good impact factor journals. So this is the first part of the this particular aspect. Next is the project proposal application for funding. So when you want to get the funding for the research or for any project, maybe program support proposal project is there, or some R&D research and development sorry, research and development proposal is there, then how to prepare the proposal and how to write it and submit it. For that purpose, first important thing is that you have to identify the topic for your project. As I told that from the literature survey, Google engine is there, or various search engines are there, or just by discussing with your experts or your supervisors, guides, or your friends, or you yourself got some idea and you want to go for apply for the project, then you should select the funding agency. So there are many funding agencies, research funding agencies in India. Some of the common agencies I have mentioned here. There can be more than this also agencies. University Grant Committee, UGC is there. DST, the Department of Science and Technology is there. ISRO is there. Indian Space Organization is there. Indian Council of Agricultural Research is there. Indian Council of Medical Research is there. Environmental and Forest Ministry is there. Atomic Energy and Regulatory Board, AERB is there. Aeronautics Research and Development Board is there. Then Research in Nuclear Science is there. Council of Scientific and Industrial Research is there. CSI is there, very common. Defense Research and Development DRDO is there. Department of Atomic Energy is there. Department of Biotechnology is there. So such research agencies are there. It is very easy to apply. If it is very easy to get the information about this just on Google also, if you put the University Grant Commission project proposal format, you can get. It's just you put name of any of these agencies on the search engine, put the project formats, you will get. Download those project formats and you should go. You should these project formats are very easy to fill up. So when you fill up the project formats, then after that this project format there in that guidelines are also given how to submit, what are the last date, in what way, in how many copies you have to submit, like that. All instructions are given. So here I have given one example. I have taken one example, and that is the project proposal application for funding, Department of Biotechnology, DBT proposal. Now, this is the DBT format, Department of Biotechnology. Their pro forma is there. What I have done, in the different pro forma, there numbers are there, 1, 2, 3, 4, 5, like that pro forma sections are there. So this is pro forma for submission of project proposal on research and development, program support. So you have to select whether it is for research and development of program or program support is there. So here part 1 is general information. The name of the institute, university, organization submitting the project proposal. Here I have given, if it is from your college, government first grade college, Bengaluru. State, which is state Karnataka. Status of the institute. What is the status of your college? especially 2F12B status, UGC approvals. Affiliate to this is affiliate to Bengaluru University, AICT and AICT, New Delhi. RUSA funded is there. It is reaccredited. You have done reaccreditation from the NAC and UGC approvals also. What is your status? That you have to mention. Name the name and designation of executive authority, which executive authority of the institution is the principal. So you have to write the name and details of the your principal. Then project title. Here I give some examples. The development of liquid bioinoculant of endophytic nitrogen fixing, phytohormone producing, and fungal antagonistic bacteria from banana species, and its evaluation against Sigatoka disease causing Mycospirilla musicola species. Means what here in this particular project, what is expected that from on the banana, the common disease is there. There is the yellow Sigatoka disease. Yellow spots are produced and drying of leaves is there, and that was great losses to the farmers. And the causative agent is the fungus Mycospirilla musicola. You want to control that fungus as well as you want to give good health to the plant, and you want to get the good crop of the banana. For that purpose, what you want to isolate bacteria from the plant, banana plant, diseased as well as healthy plants. That is the Musa species. And these bacteria, they are obtained from the plant body. So they are called endophytic because inside the plant body, endophytic bacteria. And those bacteria will be isolated, and they will be studied for nitrogen fixation. They will be studied for the PGP production, that is the plant growth hormone production, like indole acetic acid, gibberellin production. And in addition to that, it produces some compounds which are inhibitory to the fungus like Mycospirilla musicola. With three important properties in those bacteria: nitrogen fixation, hormone production, and antifungal activity. Such microorganisms you will isolate, and from then you will select the best possible promising isolates. You will prepare the mixture that is called consortium, and that consortium you will try on this Sigatoka causing organism, and you will perform the plant studies, especially the farm studies or the pot studies, and then you will see that your how this consortium works. And that will be the good thing. That this is your project. This is your project. And now in this project, then other things are there. Category of the project is research and development. Specific area is agri-biotechnology. The duration of project 3 years. Total cost 10 lakhs. Say for example, I have given here. Here these points are very easy to fill up these points. Is the project single institutional or multi-institutional? Say for example, single, say your college, first grade college, Bengaluru. Single institution is there. If you are collaborating with some other colleges or other institutions, then you have to mention. If the project is multi-institutional, please furnish the following. So as it is not, so we have mentioned NM is not applicable. Like that, on this page you can write. Next is you have to write scope of your project. Then I have given the points. So it is you will isolate nitrogen fixing bacteria. You will prepare liquid in bioinoculant as a mixture. Consortium, liquid bioinoculant will be there, and it will have property of the nitrogen fixation as well as hormone production and as well as antifungal. It will control the Sigatoka disease organism. So in 1 to 2 paragraphs, you can elaborate all these points. I have not elaborated this, I have written the point, but when you prepare proposal, you have to elaborate the point like this. Then the processes which are there. What processes are there in your proposal project? Say microbial studies are there, isolation of Sigatoka pathogen, that is the Mycospirilla pathogen. Nitrogen fixing bacteria you will isolate. Growth promoter producing bacteria you will isolate. You will isolate bacteria producing antifungal compounds. So you will try to get the organism which will have all the three properties together. Such will be the promising organism. Then you prepare consortia. Then you will formulate those consortia. You study the efficiency of consortia, whether they work or not, consortia. And then you will study shelf life of the consortia. That when you store it, preserve it, for how long the efficiency is maintained. And then cost economics also you study that, is it economically feasible or not. Like that, you have to do that, and you have to say each point you have to elaborate. And you write then project summary, not to exceed 1 page. Use separately. This is project's summary. Total your project will be like this. And in that, just the points which I mentioned, all these you will write and you will prepare project summary. Then about the investigators who are there. Then if one investigator or more is one is there, you write about him. If more than one investigator is there, then principal investigator and then co-investigator one, co-investigator second, third, like that. And again, if investigators are from your own, if single institution project is there, no problem. But if multiple institutional project is there, then some your investigators will be there from other institution like that. Then you have to give the details. So if the principal investigator is there, then name you have to write. Say X is there from your college. Say botany person is there. This project is principal investigator is botany person. Then co-investigator is you are taken as some microbiology work is there. Y person is taken from say micro department of your college. And then third co-investigator is there because some chemical aspects are there, estimations are there. Then chemical aspects are there, analysis is there. Then chemistry person you are taken from your college Z. And then you have to give all the details, date of birth, designation, department, institution, address. Every detail is to be given. Use separate sheet for if more investigators are used. So depending upon that, you have to write the details about the investigators. Then the technical details, they are very important. Introduction, 2 pages, not more than 2 pages, 1,000 words. Introduction about your subject. In that, origin of proposal, how you got the idea, how you got idea about this particular project. Introduction, 1 paragraph. Then 17.2, rationale behind this particular project. Cite the literature in brief, 1 paragraph. Hypothesis, aims and object hypothesis. You write, say 1 paragraph. Then key questions about banana production and difficulties in banana production. Some questions are there. You mention those questions. Current status of research and development in the subject, both international and national scenario. Means you have to write literature survey, national scenario and international. Quote few references. The relevance and expect out what you expect in this proposed study. You should say, for example, I have written expected inoculum and importance. Then you have written importance of the inoculum. 1 paragraph. Preliminary work done so far. Have you done some work before on this particular project? Then you should generally, when you go for any proposal, it is expected that you have some expertise, you have background of this. So you should work say 6 months or 12 months study and how it has helped to get idea about this project. So you can write 1 paragraph like this. You should write the introduction and this technical aspect of the project. Specific objectives you should write. Objectives, aims and objectives. What we say in the bullet form you should write. Should be written in the bullet form and bulleted form. A short paragraph indicating the methods means aims, objective followed by the methods. So that should be written. Say objective I have mentioned here. Isolation of that particular organism. Then isolation of the natural fixing endophyte. Isolation of hormone producing. Isolation of the inhibitory activity producing, antifungal activity producing. And then you will study these organisms, their production, extent of production of these compounds. Then you will prepare the consortium liquid bioinoculant. In that liquid bioinoculant, you use some supportive material like say glucose, some glucose amount you would add it, and you prepare the consortium preparation, and that consortium efficiency you will test in the banana plant, healthy plant, then disease plant. You will infect with the organism, and then side by side you will challenge. You will inject the consortium as well. You will perform the pot experiments and farm experiments. So this will be your aims. Common aims are there, and then you write the methodology. The collection of banana plant material from different sources, from different areas. You collect banana, say from different states. If you select your work area is from say Maharashtra is there, Karnataka is there, or nearby states are there, you can select. Or depending upon how much is the your scope, you can select the area, or you can select some districts also from the Karnataka also. Isolation, characterization, confirmation. So the causative agent is Mycosporidium musicola. For that particular Sikatoga disease, you have to confirm it. And common method is the you are biologist. Cox postulates are used in this case, and that can be done. And this is a method for isolation of the organism producing these plant growth promoters, nitrogen fixing, and different agents for the nitrogen fixation, nitrogen activity. You will perform that methodology there. Inhibitory activities you will study. then you will identify the promising isolates. You will identify bacterial isolates. You will isolate by identify by the genetical methods. The common method is the 16S rRNA gene sequencing method. If it is a fungal, you can use the 18S rRNA gene sequencing method, and by that way you can identify this fungal pathogen, Mycoplasma musicola. You can identify by that 18S rRNA gene sequencing. By bacteria, you can identify on the basis of 16S rRNA gene sequencing. Then consortium development method, plant growth promotion studies, and then after when you inject in the plant and when you get the results, then from plant you have to re-isolate the bioinoculant organism. You have to identify that yes, organism is the organism which was inoculated. In this way, you have to perform experiment. For Cox postulates, you have to confirm very carefully, and detailed methods are to be given. And this is the timeline you have to give. Timeline, the work plan and timeline. How the first 6 months, 12 months, 18 months, 24 months, 30 months, 36. Your project is of 3 years. Then the aims and objectives which I have enlisted. First, it is given in the first 6 months you will go that. In the next 12 months, 6 months you will go with this. Then all these points you will add that what kind of the achievable targets should be there in that particular time span that you have to give in detail. Then the name of the address of high experts in the field, the people who are from that field, because the funding agency may refer to these people about your project, or may send you a project for this particular purpose, about your suitability of project. I'll give some of the examples. Known people, they are there. Dr. Kapanis is there, Dr. Bhima is there, Dr. Madhamar is there, and Dr. Dayanand is there. So many people there. You must be knowing Dr. Dayanand. He's from Bengaluru. He's currently, I think, Vice Chancellor of that university. All these people who are the mainly they are in the field of Dr. Kapanis is a botany microbiology person. I myself is a microbiology person. Bhima is from botany microbiology person. Dr. Madhamar microbiologist. Dayanand is also from microbiology. So these people just you have to give their details as per the table. Then the budgetary aspects. So first is you have to prepare proper budget. 10 lakh rupees you want. Then non-recurring and recurring aspects are there. Non-recurring equipments and accessories if you want. Then say for example, I have written that for this project I want some UV spectrophotometry to analyze the chemical analysis. UV spectrophotometric methods which are there for that project. So I have mentioned that about 3 lakh rupees will be required for that. Then the total is total is 3 lakh rupees. Recurring, which is there, say manpower. That is the B1. So I just I told that we want one research assistant, which we don't have in our college. Say for example, one research assistant for collection of the samples and for field visits and so on, and for health purpose. And how much roughly I have mentioned that 10,000 rupees per month I'll be. So 1,20,000 rupees for that. So this 3 lakhs will be for instrument, for the manpower, the B1 component, 1,20,000. Then chemicals, glassware, labware, miscellaneous consumables we call. Then in the chemicals, you have to give the list wise, name a list of the chemicals and quantity also. So in first year which chemicals, in second year which chemicals, in third year which chemicals. I mentioned the amount that in the chemicals I'll require chemicals of 2,50,000. Glassware also, what kind of glassware listing should be given first year, second year, third year. So I mentioned 1 lakh rupees. Glassware, labware, lab material will be there. 15,000, 15,000, 20,000, 50,000 I require. Miscellaneous amount which will require, which is other than this. Then again 80,000. So it becomes 4,80,000. So B2 is 4,80,000. B1 is 1,20,000. Instruments is that is A part is 3 lakh, and then B3, B4, B5. B3 is traveling. You will go for. I told that you will go for sample collections and so many traveling aspects will be there. 50,000 rupees you ask for that. The first year mainly traveling will be there, and the remaining part will be the work. 50,000 contingency amount. So for example, you want for stationery there, xeroxing there, typing is so many things are the sundry aspects which are there. For that purpose, you will need, and 50,000 I mentioned that in the 3 years I require. Then overhead it is not applicable here. Subtotal of B, this B1, B2, B3, B4, B5 is 7 lakh rupees, and A, that is a 3 lakh rupees. So grand total is 10 lakh rupees. So correctly you have to mention, and for every aspect you have to give justification. It is mentioned, please give justification for each head and subhead separately mentioned in the about data. So you have to give the why you want particular these chemicals, why you want this glassware, why you want this instrumentation, then where you want to go traveling. Means in that way you have to give the justification for each and every aspect. Then pro forma of the investigators details, say educational qualifications, that the common format is that institution plays degree awarded year. For every year, say three are there, X, Y, Z, three people there are for every person one page. You can give the details. Then the positions and you should write what kinds of positions you are holding presently. If you are come from different institution, then you should write first institution, second institution. The position is assistant professor, dates from to, like that. Honors, if you have got some awards you have got, you should mention to the list. Professional experience and training relevant to the if you have taken any training, say for example how to isolate the pathogen, how to isolate bacteria, how to isolate fungal pathogen, how to collect the plant material, then how to go for estimations, like that, or how to go for identification. If you have taken training, you should mention in one paragraph. And publications you have tried. Select peer-reviewed publication. 10 best publications in chronological order. You have to mention the publication. Then you have to mention books if you have written some books, research papers, reports, patents if you have, you mention the patents. This your details you have to give, details of the investigators, and then is declaration is there. Conditions are there. They are always there. Say this research proposal when you are doing, it is not a duplicate work. It is the genuine work, original work is there, or you are not applied somewhere else for this particular, or the manpower and whatever infrastructure which will be required. That undertaking is to be given by the executive authority of your university. The principal will give that yes, I will provide each and every infrastructure, or if finance is required, if this project is accepted, and if the finance will be provided till the project finance received, it will be supply provided. Provision will be made from the institution, like that. So all these aspects which are there. Then regarding the patent, if patentable item is there, then along with the discussion with the particular DBT, then you will go for patent. All these aspects will be how to how you will submit the results and this progress of your work, that will be given, and whole information you have to give, and then final the details and the information given in the project proposal are to facture. This is the brief of undertakings. Declaration you are giving, certification you are giving, and then you have to it will be signed. If multi institution is there, then from other institutions that project coordinator will be there. Or but if it is from your institution, X, Y, Z, 3 investigators and your principal will sign. And in this way, you will complete this. Then they give the list of the references relevant to project. Give the list. Yes, list maximum 5 references. Top references, previous workers who have done that. Give the 5 top references. Then about the research support. Ongoing research project, if you have done some research projects in your college or department, then give the titles 1, 2, 3, 4. That helps to know your expertise, that you have previously handled some research projects. Because many times people they get funding from the universities, native universities also. In our Pune University, BCUD, Board for College and Department, University Department, that gives every year about 3 lakh rupees per person. in big colleges, they get almost to the 10, 15 people, 3 lakh rupees each. That kind of funding is provided by that university for the 1 year or 2 year projects. So such small projects you can get if you have done such projects, or some minor projects you have done, some major projects you have done, you give the title of the list the ongoing projects or completed projects if they are there. Then you have to write last 3 years projects you should write, and then again there will be signatures of the 3 people. 3 investigators will be there. Thank you. So in this way, you can prepare the project. You can submit to the. You have to submit that to the particular agency, and after submission, they will give you the feedback If they will call you for interview, and after the interview, then your project will be, depending upon their satisfaction, your project will be accepted. And once it is sanctioned, then you can start for your work. You can take whatever infrastructure and finance required from your institution, that will be disclosed later on when you receive the funding. So just in brief, because take into account the time factor, in brief, I have taken one example only, the DBT. In this way, you can have n number of funding agencies. You can get the formats of the search engines, and you can go for the preparation of projects and submission. So in this way, you can go for the preparation of research paper publication as well as the projects. Thank you very much.
Thank you very much, sir, for guiding us and the beginners of the research work on preparing the research papers. So especially from how to write the language and how the language plays a very important role in writing the research papers. And the professor has given good number of examples on each section of research paper. So, and as well as in preparation of project proposals. So which can be applied to our respective subjects. So sir has explained every section in detail with examples. So thank you very much, sir. It was very interesting and informative session. So we are very glad to have you, sir. So it was very nice. So I hope this will be adopted by everyone in your future work. So now I request the participants, if you have any queries or questions, you can put it in a chat box or you can ask directly with the sir and you can have the discussion. It is open to the discussion right now.
Yes, you can ask questions. Anybody?
Sir, actually, there was a question earlier. I'll just ask you, sir. If a journal is having a good citation and also having the Sci index, but taking the processing charges, is it a predatory journal? The question was.
No, that you have to get from the care list. University UGC has published a list. That's called care consortium.
Okay.
List is there. It should be from that. Otherwise, it is very difficult to say whether it is a predatory kind of journal. Because nowadays it is a crop. There is crop of predatory journals. So one should be very careful, because these predatory journals, they give the very high impact factors and like that. But one should not be go for such journals. So as far as possible, widely known journals, and again that to the approved by UGC. One should go in India at least. You should go from UGC list.
Sir, somebody is asking brief about the plagiarism in writing.
What they want exactly? What plagiarism?
During writing the research paper.
When you write your research paper, you have to see that if you take some material from some previous work, then change the language one thing. Don't take exactly same text as it is. Cut and paste. And when you take some material from some previous paper, you give the reference. When cite the reference there. If you don't cite, then there is a plagiarism. And when you take from your own papers also, you have to mention, otherwise it will be self-plagiarism. You have to be very careful wherever you take some material, because many times this thing happens when you discuss your results, and in many aspects the text, you take some references, some aspects, then you have to give the reference everywhere. If you take the table directly, you take some pictures, below that source, you write the name, what source you are used. So there it will not be plagiarism. Otherwise, it will be treated as plagiarism. Especially this thing happens, especially when you go for thesis writing. When you want to go for thesis presentation before submission, it is very, very important. Because in the thesis, many times this thing is observed, that the extent of plagiarism goes beyond the 20, 30, because it should be below 10 commonly, it should be below 10%. So that is very, very important, that you should mention the source.
Good afternoon, sir.
Yeah.
It was really a very systematic presentation, sir. Thanks a lot.
Thank
Sir, I just have one doubt when we are writing a review paper. So already I have done one review paper, one or two, where I have mentioned review. Is it mandatory that in the title we need to have that review has the review or comparative study, some words pertaining to that it is a review paper, or
Right,
we
right.
can just go ahead with the entire title?
Generally in the title, when you mention the title, then a review or a review of so and so. Generally, it is mentioned. But if you already published it, then you have to mention that
Yes.
it is a review paper.
Okay. So one is already published, but one I
No
am
problem.
planning to do
No
where I will
problem.
be
But generally, when
doing a
you
review.
write, if it is mentioned
Okay.
that it is a review article, review paper,
Okay.
then you need not to explain separately that this is a review paper. People one can understand directly from title that this is a review paper.
Okay. Okay, sir. Thank you.
Sir, there is one more interesting question, sir. If the research work is new and no review literature exists on that, so how can they proceed with the research work?
No problem. When it is totally new and no research work is done on that paper, it is totally new aspect. Then when you will start discussion, in the discussion you can clearly mention that there are no references available or literature available on this particular topic. Whatever findings they are there, your own findings are. No problem.
Okay, sir. So next, one more question, sir. What is H index? How to check it for our paper?
Directly you can go on the Google. You can go. You put your research paper paper titles in that. When you get the H index, when you on Google, you get H index or Scopus or other indexes, I index. Then you open that. In that you put your paper. When you put your paper directly, you get the citations. It is very easy. Directly you get it.
What is the difference between I index and H index, sir?
I index and
H index.
that is on the basis of the sources we have prepared that. That is the only. The agencies, they are prepared I index and H index, and according to their name, they are there. If you go direct on the Google, directly you will get it. What is impact factor, how it is calculated, what are the citation index, what is the H index, what is I index, all these indices, you will get directly full information on Google. You will get it.
Okay, thank you, sir. Any other doubts, or a few more people have raised their hands?
Yeah.
Vijayanagara.
One thing just I want to mention is, sir, regarding the fresh papers, I did it one reservoir operations of simulation and modeling, where I didn't get any references. Then before submitting to the journal, just I send it the same paper with in reference section I mentioned this is a new paper with the help of the hydrological and data collected by me. I am doing it. They approved, and I presented the paper in the international conference, and now it is published in the journal also. For fresh papers, we have to mention that this is the fresh one, and what are the references for the relevancy of the subject. We have to mention in the reference section. That's it. What I did, sir.
No, in the reference section, you will not get references with respect to your topic.
Yes.
Say reference will be available. Say for example, some methodology you use. For that purpose, some references in the references, some references will be there. You can quote references.
Yes, exactly. Because when I did the mathematical modeling in reservoir operations, I did in reference section. I put hydrology, a book of hydrology where I got some
that
of the
you can.
information.
Yeah. Direct reference may not be there.
Yes,
indirect
exactly.
references will be available.
Yes.
In the discussion section, you can discuss that this is a totally innovative topic and no references are available, or scanty references are available. Then few references you can quote if they are there.
Yes, exactly, sir.
Yeah.
Thank you, sir.
Good afternoon, sir. Ushasri from Chennai. Sir, actually, I've sent one review paper for publication in a paper with ISSN number, but not
Yeah.
UGC approved. Is it valid, sir?
No. See, if you have published your paper before a care list, you say before 2017, that is approved. But once it is beyond 2017, or once that care has come into existence, that UGC list, then it is required it should be from the UGC list. That is especially important when you go for research or PhD work. Then whatever papers they are there, they should be from care list.
Okay.
So you
Sir,
see that it should be from care list. If yet not published, then no, you can withdraw your paper and publish in the care list.
Okay,
but if it is published and it is after that care list publication, then can't help.
Okay, sir. Sir, one more doubt, sir. Sir, for writing background in review paper, can I go through multiple articles and cite all the articles, all the authors?
You mean the introduction?
Background.
Right. The background is you will introduce your that subject of the paper. That is the.
Yes.
Then for background you can write, but everywhere you have to give the reference.
Yes. So I can go for more than 10 to 12 papers.
Yeah, no problem.
Okay.
Only you have to take care that background should not be more than say 1 page. That you have to take care.
Yes.
Because if you one can write 1 page, 2 page, 3, 4 pages, but that journal will not accept because the number of pages will increase.
Okay.
And costing is as per page wise. They will say that because in the instruction to authors they have mentioned that maximum say 4 pages, printed pages, maximum 5 pages. Accordingly, you have to plan.
Yes.
Yes.
Sir, I can mention the specific unique points from each journal to quote in background, right, sir?
Yeah. You have to give the reference. The reference methods I have mentioned.
Thank you.
Just you have to write number of that in the reference list.
Yes, sir. Good afternoon.
Yeah,
I have one clarification from you, sir. Sir, at the time of submitting thesis, what they will do, university will check for plagiarism. Suppose
they will check plagiarism.
Yeah, plagiarism.
It should be less than 10% generally.
Yeah, it should be less than 10%. Suppose the plagiarism is 40%.
Then you have to revise. See what happens when you prepare your thesis.
Yeah, sir.
Check
suppose
the plagiarism.
Yeah. Suppose it is 40% out of which.
Then you have opportunity. Means it will not be final.
No, sir.
You have opportunity. You can make the changes, necessary changes. Sir, because it is given where the points are given where plagiarism is there. Plagiarism is found.
Yes,
in those points, you can make the changes.
Sir, I agreed with you, but out of which 35% plagiarism is from my own papers.
Your own papers?
Yeah. 35%. Only 5% plagiarism.
Means you have repeatedly mentioned that in your thesis. So reduce the repetition.
It's okay, sir. But plagiarism checking is 40%, out of which 35% plagiarism is from my own papers. So that is the case. University will not accept that one. University will see only plagiarism. Oh, 40% is there. This one, this is rejected like that. What to do such case?
Because the only thing is that you have to change the data and reduce your repetitions of your reference.
No, sir.
Because that is a lacuna. That is a lacuna.
Sir, yeah, when I do some research, so I will publish papers, 5, 6, 7 papers. All 7 papers I put it in my thesis. So, okay. So whatever thesis I write in that content will be from my own paper.
That thing will be there in your discussion part, isn't it?
Yeah, it is. Even it is in results and discussion and this one literature, everything.
Literature survey is not taken in the plagiarism.
Okay.
The literature survey section you should not take while you are going for plagiarism.
Yeah, let us say it is results and discussion. Whatever I publish paper, same thing I'll put it in the thesis.
Then you will not see when you in the results section, results and discussion section is there. When only 7 references are your name, say XYZ name is there, it will not be 35% of your total plagiarism.
Sir, everything, one mathematical formulation, everything is from papers only. My own published papers only. Whatever I published, everything I will put it in the thesis.
Oh.
That's the case. Plagiarism will rise up. Then university, why university will not consider those publications? They will reject a paper, reject a thesis, just
No,
by
then,
looking 40%.
in that case, you have to write to university.
Yeah.
Sometimes vice chancellor, in his capacity, if such special cases are there,
Yes, sir.
then he has power. So he will appoint a committee.
That's the main.
Your subject chairman, this
Yeah.
BOH chairman. He
Yeah.
can also, or the BUTR chairman who is there,
Yeah.
authority who is there, they also can see that, and if the fact is there, then even it is allowed.
Yeah, that is the one of the reason the students are refused to publish the papers. They will publish 1 or 2 papers, they will stop.
No,
And
do one thing. Otherwise, see, do one thing. Reduce your papers. Ultimately, what you want,
that's what it is.
submit your thesis.
Yeah, that's what it is
In
happening.
place of 7 papers, if you put two papers, doesn't matter. Along with your all papers, if you want, then you have to approach to the authority of university, and you should get special permission for that. It is allowed.
Yeah.
Because this is a special case for you, because you have so many publications, and that too your whatever present work you have done. One thing is that if that your publication is from your research, that PhD work, at the end you give the list of this thing, isn't it? Present publications. Hello.
Thank you, sir.
Hello. 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 I am Dr. Gauri Shankar from Odisha. Sir, namaste, sir. Actually, excellent discussion and very nice presentation by you, sir, on the scientific aspects. Sir, actually, I have 2 questions. One is, for example, one journal is in UGC care list, but it has it has neither Scopus nor Web of Science. Whether if one of paper I will publish in that journal, whether that will be accepted or not?
Definitely accepted. It will be accepted.
Sir, okay.
It is from
for second question, sir.
Care list.
Okay.
If it is from care list, no problem. Or LCWare.
Okay,
or
sir.
Scopus,
So
no
another
problem.
question. So sir, another question.
Yes.
Sir, for example, one journal is in UGC care list, but it has only Scopus, but not Web of Science. Whether, sir, it is
Doesn't matter.
accepted?
Yes, it will be accepted. If
Okay, sir.
it is in the list of care, no problem.
Okay.
Okay. Hello.
Hello. Hello.
Yes.
Sir, actually, this is Dr. Murli from Government Engineering College, Bangalore.
All right.
Sir, my question was. Hello.
Yes, sir. Please.
Could you able to hear, sir?
Yes,
no
you are
problem.
audible.
Yes. Yeah,
Audible,
audible.
sir.
Yes.
Sir, actually, one of my student is doing PhD, but we have scrutinized his paper for Springer Journal for the whatever plagiarism aspect. In some software it is showing 15%. In some software it is showing 10%. How it is possible, sir?
No, that kind of flaw is there in the softwares. What software is used by those Springer people, and what softwares which are used by we people in the universities?
Okay, but generally, we use.
But one thing is that for that purpose, the instructions of authors to the authors, what is given by that Springer journals, you have to follow. Their software you have to follow.
But they didn't mention any software like that. But generally, they say the plagiarism should be less than 10. They say.
No, but if they mention that in your paper 15% plagiarism is there,
yes, sir.
And if they have mentioned the points of plagiarism, that this point, then the only way is there, you can reduce those points. If they are not mentioning the software.
Okay.
Yes, anybody?
Yes, sir. Hello, sir.
Please.
Can Department of Biotechnology offers project to the physics researcher?
Physics person?
Yeah.
Yeah, definitely. It is a multidisciplinary subject, na? Interdisciplinary, multidisciplinary subject. You can apply. Hello. Hello.
So thank you. So thank you everyone for your queries. Sir will be very happy to answer your queries. So we will be sharing sir's email ID later in the groups.
Right.
So I think it is the valedictory address. It's the time for the valedictory
You can
address.
give my
It's over
email
to
address
our
to
IQAC.
people.
Yes, sir. Yes, sir. Later we will share your email address, sir. If they have any queries or questions,
right,
so they can contact you through your mail. Yes. So now it's the time for the valedictory address. It's over to our IQAC coordinator, Dr. Shubha Madam.
Right. Thank
you. Good afternoon, all. With a brainstorming session, actually we are concluding. Respected Dr. Pathade sir, today's resource person, our ever-inspiring principal, Dr. B. Chandrasekhara, organizing secretary, Shubhayam, coordinators, conveners of virtual FDP, and delegates from across the nation and abroad. I am really privileged to present valedictory address on this final day of virtual FDP on innovative teaching and research in science, exploring the new horizons. This is an opportunity to acquire knowledge on current technological research, academic developments in relevant fields, and promote the professional practices relevant to current education system. This may motivate the faculty to achieve competent teaching and learning environment, thus channelizing the development with respect to academic qualifications and personal matters. After 5 days of knowledge sharing sessions, I hope all the delegates are going home with a bag full of information which will benefit faculty and institution in particular and students at large. I think the most inspiring entity was the fruitful interaction at the end of each session, which has added to the success of this FDP. 1,345 delegates have registered for this FDP from all parts of the nation and from 3 countries also. Sessions were aired on a Zoom platform and also on YouTube live streaming. FDP commenced with invocation by Ratnakumari B.M., Assistant Professor of Botany, Professor Shubha M., Associate Professor and HOD of Chemistry and Organizing Secretary, presented the inaugural address on 10th June. On day 1, that is on 10th June, we had an excellent informative session by Dr. Chetan, Deputy Director RPPC on interdisciplinary approach to research and teaching in the context of NEP 2020, which covered all features of NEP, and we came to know about the multidisciplinary approach which will benefit the student community. On the second day, that is on 11th, Dr. Satish Gowda, Associate Professor, Department of Studies in Law, University Law College, Bengaluru, presented a talk on importance of IPR, which is very important for all the life science faculty as our research may result in patent. Session was very informative and Dr. Satish Gowda enlightened us on topics like copyright, patents, trademarks, etc. On the third day, Dr. B.L. Muralidhara, Professor and Chairman of Computer Science, Bangalore University, was our resource person, and topic he spoke was on ICT education, its relevance during COVID-19. With very apt illustrations and slides, he addressed the delegates. His talk was really enlightening. On day 4th, that is on 14th June, we had a wonderful and perfect session which suits all of us, and it was the need of the hour, and that was a session by Mrs. Rajalakshmi Kaushik, Psychological Counselor, Air Force Station, Bengaluru, on the topic, stop the stress before it stops you. It was like a counseling session for us rather than technical session. After her session, I hope all of us now are capable of handling present situation of hybrid teaching effectively and efficiently. And also we came to know that we have to have stress to some extent to perform better and from the perspective of institution also. I hope all of you are de-stressed now. Today is the final day of our FDP and we had a very informative session by Dr. Pathade, ex-principal Haribhai V Desai College, Pune, and author of more than 150 research papers, and delegates, I'm sure delegates are definitely got additional information on publication. And also I hope that all of you will start your work on publication and write projects proposals after this FDP, maybe by today evening only, and you will be definitely succeeding in this venture if you adopt the information and instructions given by Dr. Pathade sir. All the very best to you all on this endeavor. Online platforms are carefully and efficiently handled by Dr. P.G. Maheshwari during this virtual FDP, who has made it without any technical glitch throughout the FDP, which has added value to the success of this FDP. At the end of each session, Dr. Jisha, Associate Professor of Chemistry, was posting attendance and feedback, which motivated us to make this FDP more fruitful. The flyer, everyday slides, which were put on the screen and were designed diligently by Soumya, Assistant Professor and HOD of Zoology, and Dr. Shailaja, Assistant Professor of Mathematics, with inputs from all the members of organizing committee. From day one, members of organizing committee have put a lot of efforts to make this virtual FDP as most fruitful and memorable one. We are really fortunate to have Dr. Chandrasekhara as our principal, who has made this virtual FDP dream come true with all his motivation and guidance. With these words, I would like to congratulate all our delegates for successfully completing this virtual FDP and taking home a treasure of knowledge for your next line of teaching and research in science. And I hope the FDP has explored the new horizons in research and teaching in science. My sincere thanks to organizers for giving me this opportunity and being a part of this virtual FDP. Thank you, one and all.
Thank you, Dr. Shubha ma'am. So for briefing about this 5 days virtual faculty development program. Now it's the time for the feedback from the participants. The very first I would like to call upon Mrs. Alia Naz, Assistant Professor of English from National College, Basavangudi. So madam, you can share your views. Over to Mrs. Alia Naz.
Ma'am, please raise your hand so that we can unmute you. Yes, ma'am. Please unmute yourself and give your feedback.
Yeah, am I audible?
Yes, you are audible, ma'am. You can continue.
Good morning, everybody who are all present in this virtual FDP program, and congratulations to the organizers for making it a grand and successful event. I, Alia Naz, would like to thank, and I am really happy to be a part of this FDP as a participant or a delegate. And it is my privilege to have attended this wonderful session through all these days. And I was not expecting these sessions to be so enriching and knowledge gaining. I would like to congratulate once again to the organizers for curating the topics. They were very well selected and they were apt to the present times. It was like giving us the holding hand to every session it had and uniqueness. It was incredibly moving and effective because with the day one, we had to understand what is this approach towards research and teaching in the context of NEP. I like this integrity of these topics here. And at the same time, the importance of intellectual property rights was also very well explained. I think we got a lot of insights on this topic. It was really excellent. And COVID times has brought a lot of things at one shot. especially online teaching. And ICT was one such topic which helped us to bring lot of insight into that. And day 4 was like icing on the cake because we are all going through so much of stress. I personally lost my mother during this COVID pandemic, and I was really going through the stress. And this session really helped me out to manage myself. And I think this was inspiring and useful. I will only not be, getting this knowledge of how to manage stress, but I would share with all my colleagues and friends as well. So that was really good. It showed immediate results and effects actually after the tips what Madam had shared with us, especially Mrs. Rajlakshmi Madam. And then definitely today's topic about writing and publishing research papers and project proposals by Dr.G.R.Pathade sir was excellent. He gave us all those minute details of how to do and go about it. So I think it was the experience and expertise of the trainers and speakers, how they put across their knowledge and they shared with us. So I'm really thankful to all the speakers and the organizers who gave us an opportunity to attend it. Actually, if Dr. Shubha Madam from the IQAC department, if she hadn't shared this, I wouldn't have joined it maybe. So thank you, Shubha Madam, for inviting me to be a part of this FDP program. And I really thank everybody once again for giving this opportunity to give a feedback. Congratulations to the team and organizers of this FDP. Thank you, everybody. Have a great day.
You're welcome, Alia Madam.
Thank you, Madam. Thank you for your encouraging words. So now I would like, so many people have raised their hands. So Maheshwari Madam, you can unmute one by one.
Okay. Dr. Murli Mohan, please unmute yourself.
Hello.
Yes, sir, you are audible.
Good afternoon, one and all. This is Dr. Murali, Assistant Professor from Government Engineering College, Ramnagar. So I'm from technical side. The first day I got the message to attend some FDP. One of my friend from Government First Grade College. I was very interesting to mingle with science fraternity and as well as generally the non-technical people, no. But Avesam, you did a very nice FDP. It's not less than these technical people. The whatever the contents and the sessions and even the way the sessions which it has been admitted was wonderful. I'm very thankful to the organizers and all the coordinators who have been supported to successful for this entire event, and congratulate all the coordinators and thankful for giving an opportunity to attend this wonderful FDP. Thank you, one and all.
Thank you, sir.
Benazir A.
Ma'am. Good afternoon to all. I am Benazir, Assistant Professor of Mathematics, HKRH College, and a research scholar from FDK College. It's my pleasure to share my thoughts in this occasion. Hope this webinar had inspired everyone. I really feel privileged to be a part of this webinar series. All the sessions were extremely good and informative. I thank the organizers and resource persons of this webinar for updating everyone about teaching and research. It was very helpful to reduce stress and uplift myself with positive thoughts. I assure that this webinar had created a huge impact on us. Once again, I extend my sincere gratitude to the organizers for arranging this 5-day program in this pandemic. Congratulations, organizers. And especially I like to thank Dr. Subha Madam to giving this wonderful opportunity. Thank you. Thank you, one and all. Thank you, ma'am.
Thank you, madam. The next.
Thank you.
Ramkrishna Reddy, sir, you can unmute yourself. Please unmute, first unmute, sir.
Yes, thank you. Thank you very much for giving me an opportunity. So other participants already, they have mentioned about the topics and resource person. I need not once again repeat that. But anyhow, thank you very much for giving this opportunity. One is topics are very excellent, and resource persons which are selected also done their justification. So that is only I can say. Another important aspect is that many other webinars also I have attended. The technical issues were not there. That is more important point, and I appreciate and thank all of you. And another important point is coordination is more important. I have also organized. I am from Kalburgi, Sharanabasweshwara College of Science, Kalburgi. We are also organized, and I have attended many programs. Wonderful coordination. That is one point I would like to say. That is the main key for the success. And the time management. Entry as well as exit also. Of course, exit sometimes it may be, and the interaction based, that is. And the topics were very good. That is why interactions was also very good. That's what I want to mention, these main important points. One is coordination of all of you, and technical errors were not there, and the time management. These are the three important aspects. Otherwise, it is very wonderful. I thank Principal Sir, then Shubha Madam, Dr. Shubha Shishikant Madam, and Shailaja Madam, and all the team of FDP on my own behalf and the behalf of all the participants. I thank you. Thank you very much for giving me an opportunity. Thank you very much.
Thank you, sir.
Dr. Lata.
Can you hear me?
Yes, ma'am.
Okay. Everyone has spoken very nicely about the FDP. I also are of the same opinion. Whatever points they have said, I also second all that. I am with them. But one thing I would like to tell is, if I had not attended the FDP, I would have lost quite a bit of the information what was provided. Especially yesterday, Rajalakshmi Madam, she was too good and it was very effective. And most of it, at least we can forward it to our students when we take classes. And once in a way, in between the classes, this will be a slight deviation for the students, which will really help them to do their studies well and also come up in their life. Thank you. Thank you, organizers, for the excellent job you have done. And I think in the college, I know Shailaja Madam very much. She comes for valuation. Thank you, Shailaja Madam. And excellent job done by every one of you. Thank you. Thank you.
Thank you, madam. Thank you for sharing your feedback. So one more person. Yeah. So I hope we will proceed further. So I thank very much for sharing your feedback. So actually, these words are the boost for us to organize this kind of programs in future also. So thank you very much, all the participants for sharing your feedback. So now it's the time for the presidential address. So our principal, though he is busy for his administration and other work, so he is with us from the morning. Now I request our beloved principal sir, Dr. Chandrasekhara B, to address the gathering. Over to principal sir. Sir, please.
Thank you, Dr. Shailaja. Good afternoon, respected resource person. Today's resource person, Dr. Pathade sir, all the participants, Dr. Shubha, IQAC coordinator, Professor Shubha, convener of this 5 days national FDP, all the HODs and faculty members of the science departments, dear participants. I feel myself privileged to share few of my thoughts on this occasion. As we all know that great things are never done by an individual. Rather, they are done by a team of individuals, a group of people. That means this FDP has proved that it's a great team effort, the teamwork they have done. I congratulate all the members who are involved, all the faculty members who are involved in organizing this national FDP. I believe in an investment in knowledge pays the best interest. That means there should be a takeaway when we attend programs like this as such. I hope all of you have a takeaway from this FDP. An expenditure of words without income of ideas will lead to intellectual bankruptcy. I hope there is no bankruptcy here, and every word expressed by the resource person contributed a lot and definitely enriched the knowledge of all of us who involved in this FDP program. And this FDP definitely help us to acquire new knowledge and diverse skills, because today the diverse skills we need to acquire to manage diverse students. We have students with different cultural backgrounds, different traditions, religion, caste, creed, background, nationality, etc. In order to manage the students with different skills, diverse students, we need to acquire diverse skills. They are very, very important to be a good teacher, a good administrator, etc. So today, I hope this FDP definitely brought some attitudinal changes amongst us. Definitely, a right attitude is definitely important. Always, I believe in a positive attitude, always find a solution. Negative attitude always creates problems. And definitely, we all move towards the positivity in bringing by attending this FDP as such. And definitely, it changes our attitude towards teaching as well as research. And on the whole, a whole teaching, the pedagogy we can adopt, and there is a change in the teaching behavior of teachers we could definitely notice by attending programs like this. And also we could acquire a new abilities. Definitely we need to acquire an abilities to manage multiple roles at a time, and also a new responsibilities we could assume. Many times we are reluctant to adapt or accept the new responsibility, and only because of the dearth of knowledge or lack of knowledge. When we have enough knowledge, a new skills, and all these diverse skills which are required, definitely we can come forward to accept any responsibilities. The responsibilities are the roles we could play effectively and efficiently in any of the institution. So definitely I believe in there is an integration of technology and teaching with this FDP as such. And it also helps us for a professional development, and like teachers, researchers, and as administrators, all of us can definitely improve our professional the abilities. And there is a career development opportunity here. Career development also it is useful, where many times where we can use this FDP certificate for our career advancement, I believe. And also there is a personal development through attending programs like this, and we would effectively plan our activities effectively, and also we could acquire interpersonal skills and also an effective communication skills as such. And this FDP definitely gave us an opportunity, all of us, to an opportunity to learn, to acquire new skills, new abilities, new techniques, new methodology, and altogether a new approach to write research papers, and also many research proposals as such. I hope all of us now, many participants can have a make their job easy to prepare writing research proposals and also the research articles. And they can publish many of the research papers and research articles in days to come. I also believe there is a there is a financial if this online platform is not available, probably would make all the 1,000 people, 1,000 participants and to assemble in one place and conduct programs like this, that too for 5 years with a little amount of expenditure, with a meager amount of expenditure, we could able to organize programs like this. In turn, it benefits hundreds and thousands of participants as such. And we could also learn a proper session design. We can definitely design our sessions effectively in a systematic way. And with that, definitely we can have a better student-teacher relationship. That is very important for a teacher, and that's what where we believe in that we should love the students like our own children. Then only a teacher can definitely bring the some merits for his job, his profession as a teacher as such. On the whole, I can conclude by saying that definitely this FDP paved the way for new approach to research and also for preparing project proposals. With this a few words, and I appreciate the participants who have given wonderful feedback. Definitely, if there are any lacunas, if there are any shortfalls, and in the next future course of time when we arrange programs, definitely we can improve further. With this few words, I conclude my speech. And again, once again, I take this opportunity to congratulate all the professors like Dr. Shubha, IQAC coordinator, Professor Shubha, convener and the HOD of chemistry department, of the convener of this FDP program, Dr. Venkateshappa, convener of science forum, and all the HODs and the faculty members of all the science departments for taking this FDP to a grand success. So thank you very much. With this, the concluding remarks, and over to Dr. Shailaja.
Thank you very much, sir. Thank you for your encouraging words and the encouraging presidential remarks. It's over to Ratnakumari Madam to propose the vote of thanks.
Thank you, ma'am. The best and beautiful things in the world cannot be seen or even touched. They must be felt by the heart. Expression of gratitude is one such good thing. A very warm and cherished afternoon to everyone. I, Ratnakumari B.M., Assistant Professor from Department of Botany, consider the occasion as an honor and privilege to extend the vote of thanks and acknowledge the contribution of those who worked hard to make this 5 days virtual faculty development program a grand success. To begin with, I would like to express my heartfelt gratitude to our today's resource person and an eminent speaker, Dr. Pathade, retired Principal Haribhai V Desai College, Pune, for accepting our invitation and for delivering a lecture on writing and publishing research papers and project proposals. On behalf of Science Forum and GFGC Vijayanagara, I thank Dr. Pathade sir for his excellent, most remarkable and highly informative. I'm overwhelmed to put forth my deep reverence to all our esteemed guests and resource persons of all the 5 days faculty development program. So on behalf of GFGC Vijayanagara, I thank Dr. Chetan Singhai, Deputy Director at RTPC, for an extraordinary information on the topic, interdisciplinary approach, research and teaching in the context of National Education Policy 2020, which was held on 10th of June. I would like to thank Dr. Satish Gowda N, Associate Professor, Department of Studies in Law, University Law College, Bangalore, for his most valuable information on the topic titled Importance of Intellectual Property Rights, which was held on 11th of June. I'm also very grateful to Dr. B.L. Muralidhara, Professor and Chairman of Computer Science, Bangalore University, for an enlightening thought on the topic ICT education, its relevance during COVID-19, which was held on 12th of June. I would like to acknowledge our gratitude to Mrs. Rajalakshmi Kaushik, Psychological Counselor, Airport Station, Bangalore, for the very effective session, which was held on 14th of June on the topic Stop Stress Before It Stops You. Any academic activities such as this dimension can happen only with a dedicated staff who are well guided and advised by the most able personality who needs no much introduction. It's none other than our magnificent principal with the constant pursuit for excellence in all aspects. Our principal, Dr. Chandrasekhara, who stands as a pillar of power with integrity and organized vision. So with deep sense of appreciation, I thank our principal on behalf of Science Forum. Thank you, sir.
Thank you.
Well, such an event cannot happen overnight. We'll start rolling around a week ago. It requires thorough planning and execution. We have been fortunate enough to be guided by the team leader and supported by the team of very motivated and dedicated colleagues who complete the task successfully. Hence, a sincere thanks to the organizing secretary, Subha M, HOD Chemistry, for organizing such a meaningful, useful faculty development program very systematically and effectively. And I thank all the team members of the Science Forum for their untiring efforts in making this event a most memorable one. I also thank Dr. Shubha, HOD Botany, and IQAC coordinator for the validated address and report of virtual FDP, and also for the constant support in the organization of this program. I'm further thankful to the convener of Science Forum, Dr. Venkateshappa, HOD Physics, for the constant support and encouragement in the organization of this program. On behalf of Science Forum and GFGC Vijayanagara, a big round of applause and lots of appreciation to all the participants who made this FTP a great success. My special thanks to these participants, Professor Aliana, Assistant Professor from Bangalore, and Dr. Murli Mohan sir from Ramnagar, and Mr. Benazir, Assistant Professor, Department of Mathematics, and all Ramakrishna sir and Professor Lata, who all shared their wonderful experience and appreciation today. And this feedback will keep us motivated to organize many more such programs. Thank you, participants. From the bottom of my heart, I thank all the moderators, technical team led by Dr. Maheshwari, HOD Mathematics, and Dr. Jita, Department of Chemistry, Dr. Nazia Hassan, HOD Computer Science, and Professor Somya, HOD Zoology, who supported to execute this activity without any technical problem. Last but not the least, I want to express my heartfelt gratitude to everyone who has supported us in the successful completion of the 5 days virtual faculty development program on innovative teaching and research in science, exploring the new horizons. To conclude, let me once again reiterate my gratitude to all. Stay home, stay safe, stay healthy.